Hello and welcome to the second part of our enzymes virtual lesson, this time looking at how pH and temperature can affect enzyme activity. The first thing to think about is what is a chemical reaction and how could you speed a chemical reaction up? So pause the video and write your ideas down. A chemical reaction is a process in which one or more substances, which are called the reactants, are converted to one or more different substances, which are the products. A chemical reaction can be sped up by a change in temperature, increasing the surface area of the reactants, increasing the concentration of the reactants, or adding a catalyst. And hopefully from the last video, some of those words like surface area and catalyst are ringing a bell. An enzyme is a special protein that breaks down large molecules into small molecules, which helps faster digestion. And a catalyst is a substance that speeds up the rate of a reaction without being used up itself. Remember that when we're talking about enzymes, we're talking about this lock and key idea, this idea that an enzyme fits a certain substrate. So a carbohydrate molecule is broken down by a specific type of enzyme. Living reactions are reactions that happen inside an organism and they're part of a process called metabolism. Metabolism is the sum of all the chemical reactions in your body. I want you to think about some of the reactions that may be part of metabolism. Some examples would be respiration, digestion, growth, and in plants, germination and photosynthesis. Almost all chemical reactions in an organism are controlled by enzymes. Without enzymes, these reactions would be too slow for life to exist. Most processes in an organism consist of many reactions in sequence, and each one is catalyzed by a specific enzyme. Remember that one enzyme does one reaction. Enzymes are catalysts. They increase the rate of a chemical reaction but are unchanged at the end of it. That means that they can be used over and over again but are not used up in the reaction. And you can see on this graph that without an enzyme, which is the blue line, the reaction still happens but much slower. And in the case of us, it would be far too slow for our lives to exist. And the red line is with an enzyme, and you can see that the reaction happens much faster. So the enzyme is a catalyst, it catalyzes or speeds up the chemical reaction. We're going to recap here and see what we can remember about enzymes, the substrate they break down, and the product that they form. So pause the video here and try and fill the table in for me. So carbohydrates breaks down carbohydrate and it breaks it down into simple sugars and glucose. Amylase breaks down starch into simple sugars and glucose. Protease breaks down protein into amino acids. And lipase breaks down lipids or fats into glycerol and fatty acids. Biological reactions are affected by different conditions just as much as any other chemical reaction. So think back to the start of this video, we said that was surface area, concentration, temperature, and pH. And enzyme structure is affected by pH and temperature, and as a result, both these factors will affect the rate of reactions. I always think of it as Goldilocks and the Three Bears. Goldilocks like things a particular way, or as she said in the book, just right. And enzymes are no different. They like a certain pH and a certain temperature. Each enzyme is different, so one enzyme may prefer a pH of 3 and one may prefer a pH of 10, and the same with temperature. But each enzyme has what we call an optimal or an ideal set of conditions. This graph is showing the rate of an enzyme-controlled reaction. And what we can see is that as the temperature increases, the rate of reaction increases. And this is because we're giving the enzymes and substrates more kinetic energy. Think back to particle theory about how liquids and gases flow around. If you increase the temperature, you're making them move much quicker because you're giving them more kinetic energy. So they're going to bounce into each other more and bounce off the edges of the container. The same thing's happening with enzymes and substrates. If you increase the temperature, they're bouncing around and colliding with each other a lot more. Now, with enzymes, we're thinking about Goldilocks and the three bears, how Goldilocks likes things a certain way, or as she said in the book, just right. You can see on this graph here that this enzyme prefers a certain temperature, and it prefers 37 degrees. This is where it's going to work the best and as fast as possible. If it's too cold, it will still happen, but it won't happen very quick and probably too slow for live processes to work. And if it happens too much, if the temperature is increased too much, the enzyme itself is going to denature, its shape is going to change. You can see a diagram here of what denatured is, and you can see that the active site of the enzyme is changed shape, and that means the substrate isn't going to fit it anymore. So if you imagine that this was a lipase enzyme and that was a lipid substrate, the lipase enzyme can't break down the lipid anymore because it's denatured. It can't fit anymore, like jigsaw pieces that have changed shape, or a key that doesn't fit a lock anymore. So it doesn't do its job, and it's a permanent change. The exact same thing with temperature can be seen with pH. The graph may look the same or look slightly different, but not being at the optimum pH, being too high or too low, again causes that enzyme's active site to become denatured, and it loses its important shape. So over here, this enzyme likes an optimum pH of just over 7, and any lower means it will be denatured, and the same with if it was any higher. Pepsin is an example of a protease enzyme. It catalyzes the breakdown of protein into amino acids in your stomach. The reaction is pH dependent because the normal pH of the human stomach is around about 1 or 2. To investigate how pH affects the activity of pepsin, scientists added pepsin to egg whites. And what they were waiting for was to see when the solution went clear, because that meant that protein was broken down. There's a table here of the scientists' results, so we can see that we have the pH of 1, 3, 5, 8 and 12, the time taken for it to turn clear in minutes, and then we have the mean, and the mean is calculating, so I want you to pause the video and to calculate the mean. If you know how to do that, pause it now. If you don't know how to calculate the mean, you add the 3 numbers together and divide it by the total number. So in the case of the first one, you'd add 8.5 to 8.1 to 7.9, and you divide it by 3. So the scientists' results were 8.2. That was because it would have given you 8.16 recurring, and we need to round it up to the 1 decimal point, which is 8.2. Then you'd have got 5.93 recurring, so round down to 5.9, and then 12.4. We can assume that the optimal pH here for the pepsin enzyme was a pH 3, and that's because the time taken for the solution to turn clear was much quicker at pH 3. The way you could test this to improve the results would be to test pH 1, 2, 3 and 4, because the results are relatively close together, because you may find at pH 2 it happens quicker. What may have happened to the enzyme at pH 8 and above? I'd like to pause the video and write down your ideas for me. So we can assume that the enzyme was denatured, and that's because there was no reaction after 15 minutes. If we're predicting that the pepsin optimal pH is 2 or 3, as looking at the table of results, then actually that's a massive difference going to pH 8 and above. So the enzyme shape, the active site will have changed, and it won't have been able to work or combine with the substrate. Okay, another activity here I'd like you to do is to compare the two enzymes using this graph. So what is the optimal pH for both enzymes? And where would you presume that these enzymes might be? So we've got Pepsin's pH as optimal at just over 2, looks a little bit closer to 2.5 maybe. And we could assume that perhaps Pepsin was in our stomach because our stomach is a strong acid. Trypsin's optimal pH is closer to 8, again looks around about 8.5. So we would assume that this isn't the stomach because actually the stomach's an acidic environment. Trypsin must be found somewhere where it's a much more neutral pH. The final task for you to do is to find out the answers for these, and this is going to be a research activity. So what enzymes are in each product and why? So biological washing powder is full of lipase and protease enzymes, and that's because they digest food off our clothing. There's an enzyme in baby food which helps to pre-digest protein to make it easy for babies to digest. So hopefully you already know the answer to that from the reason. And then finally go and have a look at slimming foods. So what's in them and why. In the next video we're going to be moving on to blood.